Okay, in the last session we have discussed how to produce the monoclonal antibodies by the hybridoma technology in connection with the generation of AbGymes, which are basically antibody acting as enzymes. Okay. Now, I ended up by saying that as organic chemist, what we can do is we can help in designing of the hapten, because it is the hapten which will be attached to the protein, the carrier molecule, and the hapten should have the your epitope. Epitope is required in the hapten. Okay. Now, the initial, what are the different strategies to come out with the hapten design? First strategy was really not transition state analog, because the simplest strategy is, if you know the transition state, make an analog of transition state, attach it to the protein, generate the monoclonal antibodies. But that was not the initial studies that were made. The initial study, by the way, this whole catalytic antibody is discovered in the western coast, in the California, particularly in an institute called Scripps Research Institute, where two scientists, Richard Lerner and Peter Schulze, they developed this technology, the catalytic antibody. Okay. So, the first examples were based on producing the catalytic site in the antibody, because the enzymes have a catalytic site. Okay. And this catalytic site contains amino acid residues that help in doing the reaction. Like in chymotrypsin, what you have seen, that in chymotrypsin at the catalytic site there is a catalytic triad, serine, then histidine and aspartate. Okay. So, if these three, if you can generate them artificially at the proper position, then you can make an antibody which looks like, which will catalyze like chymotrypsin. But to generate three different amino acids at particular at design positions is really difficult. So, people went for actually producing the active site amino acid, just only one, but which is involved in catalysis. Okay. So, the first technique that was used was basically generating the catalytic site which will catalyze the reaction. Okay. I think if I give an example, then it will be much more clear. See, there is a molecule, a nitrophenyl with a fluorine at the benzylic carbon, and then there is a hydrogen, and this hydrogen has been made quite acidic because there is a carbonyl group attached to it. Okay. We know that if I want to make a double bond here, I will definitely add a base, and the mechanism for that is that the base abstracts the hydrogen and the CH bond pair goes here and the fluoride leaves. So, that is the simple mechanism. So, it is a beta elimination reaction. Okay. Now, instead of base, now if I want, suppose this we have not specified R, because sometimes what happens, R may be some group which is very sensitive to base. So, you cannot use base in that case to induce this elimination, but we know that enzymes work at almost neutral pH. So, that is the advantage. Another advantage of catalytic antibody, that instead of base which can do this elimination, whether if you can generate a catalytic antibody which will also do this elimination process, then that will be very useful, because that will be under very mild condition. Of course, this is an example which is nothing but they wanted to have a proof of concept that yes, this is possible. You can generate the particular amino acid responsible for catalysis at the catalytic site. which is the active site. Okay. Now, what was done, that you know that if I have a, if think that if the antigen which is invading our system, suppose that has lot of amino acids like lysine, lot of amino acids like lysine. So, its surface will be full of lysine and lysines will be positively charged. Okay. Now, if this is the antigen, now if an antibody is generated against it, if an antibody is generated against it, I can definitely assume that the antibody must be such, because antibody is recognizing this, the antibody must be such that it has got lot of carboxylate groups, because only then there will be an interaction with the NH3 plus, because otherwise how there will be binding between the, or how there is recognition between the antigen and the antibody. So, if the antigen has lot of positive charge on the surface, so definitely the antibody will have lot of negative charge on its surface when it binds. Similarly, if the antigen has lot of aromatic rings, suppose, then the antibody will also have aromatic rings, because that will give you pi-pi stacking interactions. Okay. So, these are certain very simple things which you can predict that this is my antibody that will be looking like this, depending on the structure of the antigen. Now, in this case, remember the antigen what you are using is basically your that reaction substrate here. Okay. What you want is to generate an antibody which will have a, so basically what I want to generate an antibody which is a protein, instead of the base, the protein has a base. The protein has a base right at that position where the hydrogen is residing. If the protein has a base which is generated here by manipulation, then the same reaction will take place, because what you need is a base very in proximity with the hydrogen. If there is a basic group in proximity with the hydrogen, and if this molecule, of course, if this molecule binds to this antibody, that is also very important, some binding parameter has to be there. So, it binds and the base is right in front of the hydrogen. So, now the base will abstract the hydrogen and the elimination process will take place. Now, that means the what you have to do when you design the hapten, you have to think that how can I generate a basic center at the catalytic site. How can I generate? I already told you that if the antibody is positively charged, then you get a negative carboxylate in the antibody. Okay. Now, here you want a basic group. What is a basic group in a protein at biological pH? That is also sometimes we think that the basic groups are arginine, histidine, arginine, lysine, etc. But the problem with these, that at the biological pH, lysine is present as NH3 plus, its lone pair is no longer available. Arginine will be present as the positively charged conjugate acid. So, they are not bases in the biological system at the pH 7.2. So, what are the bases then in biological system? A base is something which has got excess electron, either the lone pair or it could be negatively charged, like OH minus. So, you have to think of which amino acid residue exists as the negative charge, and that is nothing but the carboxylate, because it is the carboxylic acid at biological pH, glutamate or aspartate is present as the anion. So, that is your source of base, it is the carboxylate. Okay. So, if that means I have to generate a carboxylate at the active site. How can I do that? If I have a hapten, see here it is written, if I have a hapten like this, where I put a NHMe plus here, that means the hapten have a plus charge somewhere in the middle where there was the hydrogen attached. So, I have a positive charge here. I keep this aromatic ring intact, other things more or less intact, only I have to attach the large protein, because that you know that you have to attach the carrier protein. So, you make a, this is up to this point is your hapten, and then you attach it to the protein. This is the hapten. In the hapten design, what you will notice that this part I did not change, because why should I change? Because this part will make another aromatic amino acid here, which will recognize this nitrophenyl group and have a stabilizing interaction by pi stacking, and this NH plus, NHMe plus will generate definitely a carboxylate here, because in order to have this antigen antibody interaction. Okay. And exactly that was what was done. So, this molecule was injected to mice and the antibody was isolated, the monoclonal antibody, and it had an aromatic group here which stabilized the nitrophenyl, and it had a carboxylic group right where you wanted, and this then underwent elimination reaction as desired. So, this is one of the first example of catalytic antibody, but again I repeat that this is not the transition state analog approach. This is an approach which directs the generation of the catalytic residue at the in the antibody. Okay. Now, let us go to other examples. Okay, here are the different strategies. I just told you that this is the first strategy, that generation of catalytic residues. I have given you generation of a carboxylate. If some reaction, if you want generation of a positive charge, something like NH3 plus, then you have to use a carboxylate in the hapten, just the reverse way. Okay. Then now we will go to the, I think this is all detail. So, we can we have already discussed this. So, we can skip it and go to the next one. Okay. The next one is that what is the transition state analog approach. And the first reaction that was tried was the hydrolysis of say an ester. Here is X, suppose this X is O Me say. Okay. Alkaline hydrolysis of an ester. Now, this is also called saponification, hydrolysis. We know that this is also called saponification of ester. Now, the mechanism of ester hydrolysis by base is like this. Suppose this is your base. So, the base first attacks and forms what is called a tetrahedral intermediate. Okay, this is a tetrahedral intermediate, and this is your redetermining step, this attack by the nucleophile onto it. It is a bimolecular reaction, two molecules are involved to form the intermediate via the transition state, and the next step is this O minus comes back and this the leaving group leaves. So, you get the hydrolyzed product. Okay. The hydrolyzed, this is Y, suppose the Y is your OH minus, then you get the carboxylic acid. Okay. So, this is the mechanism, that means hydrolysis of an acyl system with an ester goes through a tetrahedral intermediate, okay, via a transition state. Now, as I said, the intermediate is the one closest to the transition state, and so when I do the hapten design, I can take the intermediate and see which molecule resembles this, but is stable, because this is not stable. Nobody could isolate this. You have to, once it becomes O minus, it is very transitory, this comes back and X leaves. So, you cannot have this taken in a test tube and seal it and that will remain forever. That is not true. This is also transitory. Okay. So, what you need is a stable molecule which looks like this. That means you should have a molecule, because this is tetrahedral, which should be tetrahedral. Number 2, it should have a negative charge on the outward atoms. Okay. And basically that is it. So, what you need is your R group. I said that do not disturb the R group, because if the R group, depending on the R group, your binding parameters are decided. What you do is take a this is called phosphonate, sorry, phosphonate. Okay. See, basically what we are doing, phosphonate is when a phosphorus is directly attached to a carbon, that is what is called, let us just remove y and x. We just say that this is minus and that is your, say, OR right now, right, OR. So, this is a stable molecule, this is a phosphonate molecule. Now, look at the structure of the tetrahedral intermediate and the phosphonate. Instead of carbon, what you have is a phosphorus, but this is tetrahedral. And this has also got a negative charge. This is like the carbon CO minus, you have phosphorus O minus. So, instead of carbon, you have just phosphorus. Phosphorus is just slightly bulkier than carbon, that is the only difference. So, what you need to do? You take an example, let us see. So, this is a, where is that? This is the H star, okay. This is the H star functionality, and you want to hydrolyze it, this H star. And the normal is saponification, OH minus. So, this is the tetrahedral intermediate, and this tetrahedral intermediate, then it comes here and this group leaves, this groups leaves here. Okay. So, you get the carboxylic acid and the corresponding phenol. In this case, the phenol, the alcohol part is the phenol. Okay. So, that is the mechanism. So, this is your intermediate. So, what will be your hapten then? The hapten is this part, you do not change, again I repeat, this part remains the same. This is now, instead of carbon, you take a phosphonate, P double bond O minus, and the other part remain the same. Only thing, through this R, you attach it to a linker and then the protein. You attach it to a linker and then that goes to the protein. Okay. So, this is the hapten, it resembles perfectly the that intermediate, and the intermediate is close to the transition state. So, if you can generate an antibody which recognizes this, then that is going to catalyze the hydrolysis of this compound. Okay. So, that is very simple, and they have done that. Okay. Similarly, if you want to hydrolyze an amide, it is like all these proteases do, amide, again you can do it with alkali. So, if you add alkali, again you have the tetrahedral intermediate, and then the tetrahedral intermediate collapses and this goes out, and you get the carboxylate and the nitroamine. Okay. You want a catalytic antibody to catalyze this reaction. So, now you generate the hapten. Now, here is, instead of carbon, you have nitrogen. So, this is nitrogen. Now, this phosphorus, it should have a double bond O. Better I write it, whatever is written is the resonance form, but I just resonance hybrid form, but I am not, I write the full whatever. So, double bond O, O minus, and then this whole nitro group is there. Okay. Via this group, you attach it to the protein and then generate the antibodies. So, this is the this is what is called phosphoramidate, this type of phosphorus compounds. Okay. So, this is very simple. Of course, the reactions are not very difficult. The reactions are basically you are trying to synthesize, generate catalytic antibodies for hydrolysis of amide, for hydrolysis of ester. They have done also hydrolysis of carbonate. All these things they have done. But this is important. You might say that, sir, these reactions are quite easy. But as I said, in the actual case, you might have certain reactions, certain groups which react under these conditions. This is very mild when you use the catalytic antibody. And the other thing is that this will be, because these are enzyme like reactions, their turnover number will also be very high, unlike the basic conditions, the saponification. The turnover number is also very high because they are enzyme like catalysts. Okay. Now we come to a more difficult reaction. Okay. This is, you know, some of the amino acids, as I said, some of the amino acids are essential amino acids, some are non-essential amino acids. Essential amino acids means you have to give them from outside. Okay. Now, some of the microorganisms, they can make one particular amino acid like phenylalanine, which we cannot make. Phenylalanine has to come from outside. Okay. Now, the biosynthesis of phenylalanine is little complex, but we have taken only some intermediate steps where phenylalanine biosynthesis is involved. Okay. Now, this is a molecule which is called codismic, the spelling is wrong, codismic acid. Codismic acid. Okay. So, via some biosynthetic pathway, this molecule is generated, and this codismic acid by an enzyme called codismate mutase. It is said codismic mutase, but actually codismate, because all these, remember, all these acids are actually present in the carboxylate form. That is why we always have glutamate aspartate. So, this will be codismate mutase. Mutase basically an isomerization reaction where the skeleton changes. Isomerization means molecular formula of this is same as that. So, it must be a kind of a rearrangement reaction. Okay. So, that isomerization reaction is done by codismate mutase. Mutase means where the skeleton changes. Okay. The skeleton looks entirely different, what is there. Now, if you look at this reaction, if you want to do it chemically, you can do it chemically, but you have to heat it at a very high temperature. And a reaction that happens in organic chemistry, we do it like this, that this bond goes here, this comes there, and that goes here. Okay. This is an example of what is called a sigmatropic reaction. Sigmatropic reaction, and specifically this is called a 3,3 sigmatropic reaction. Why 3,3? Because the bond that is broken entirely is this one, means which detaches. The sigma bond that is broken, we are not talking about the pi bonds. The sigmatropic, that means we are looking at the sigma bond which is broken. If you look at all this, the sigma bond that is broken is this one, and then this will be 1 and that will be 1, and you number it on both ways. So, this becomes 3 and this is also 1, and that is 2 and that is 3. So, this becomes ultimately the 3 and 3 carbons are combining with each other. So, that is why this is called a sigmatropic 3,3 sigmatropic shift. Okay. Now, this is the mechanism. Of course, this is not the actual geometry through which the reaction takes place. The actual geometry is shown here. It's little this, the shape of this ring is like this, is like a kind of a boat kind of thing, inverted boat. Okay. And then you have this oxygen, you have that carbon, then the double bond and the CO2 minus, and here is double bond. That means this is the same molecule. But actually this is not the geometry. When the reaction takes place, the molecule takes up this geometry. And then here what is what you can argue, that suppose these 3 and 3 are quite far apart, how are they reacting with each other? But in this geometry you can there is no such problem, because I can draw it here, this let us see. So, this is the O, then the double bond, and this is the CO2 minus, and here you have a CO2 minus. So, when the reaction takes place, the arrow goes like this, you have a transition state which will look like a chair form, which will look like a chair. It is like this, which looks like a chair. I think if it is, let us see in the next slide. This diagram is not very well done. I think I will try to see, to show you that how it looks like a chair. It's actually, the problem is eraser. Yes, let's see the eraser. That is something. So, this is the your, this is the oxygen. I think better remove it here. This is the double bond, this is the CO2 minus, and your earlier there was a double bond here and a double bond there, and there was OH here. Okay. So, now the reaction goes by this, this comes here, this goes there, that goes there. Okay. So, what you have is oxygen, then this, then you have this, something like this. It's close to a chair if you look at this position, more or less like a chair. So, it's a chair-like transition state which is the intermediate chair-like transition state that is involved in this process, and that is true for all 3,3 sigmatropic reactions. They go by chair-like transition state. There are reactions which are Claisen rearrangement, that is a 3,3 sigmatropic shift. Okay. Now, earlier people thought that this is a class of pericyclic reactions. Okay. This occurs either by a heat or by light, we know all this. But now earlier people believed that pericyclic reactions cannot be catalyzed. They are unaffected by catalysts. Okay. But here is an enzyme chorismate mutase which does the rearrangement very fast, okay, at room temperature. You do not have to heat it. Okay. So, that actually broke the myth that pericyclic reactions can be catalyzed. How the enzymes are catalyzing it? Basically, the enzyme helps the molecule to adapt a structure, a conformation through which the reaction takes place. The enzyme helps the molecule to bind, adopt a conformation through which the reaction takes place. Okay. So, that is now if you want to make a catalytic antibody to have this reaction catalyzed, what you need to do is make a transition state like molecule which is stable like this. See, this is the transition state which we are talking about, okay. And this is there is a OH here earlier, remember there is a OH here in corysmic acid. So, that OH, via that OH you attach it to the linker. The linker, remember, has a diazo compound, and through the diazo compound you attach the protein, because diazonium salts are prone to attack and the diazo compound leaves. So, by that you can attach your protein part. Okay. So, basically what you have done, you have made a molecule which looks like the transition state, but this is stable. You synthesize this molecule and via this OH you attach it to the protein. Now, you generate catalytic antibody, and that antibody has been, it is very successful, that antibody catalyzed this Corismet mutase reaction, Corismet going to prephenate, right? Sorry, hey, what happened, just a second, prephenate. This is called prephenic acid. This is the intermediate for the phenylalanine synthesis. So, this is a very critical step, but this is essential amino acid. So, this has to be come from outside. The question is how the plants or the microorganisms make phenylalanine. So, this is the route. So, via the corysmic acid goes to the prephenic acid, then there is a decarboxylative, I can show you decarboxylative dehydration, but here OH is a bad leaving group. So, it has to be made to a phosphate. So, that leaves and that gives you a compound which is this one CO2H, a keto acid, and alpha keto acids. When we will read the coenzyme, some more coenzyme chemistry, we will see that these alpha keto acids can be converted to amino acids. So, that is the story of phenylalanine, and that has come because of this catalytic antibody. The last reaction is what I said, that some of the reactions which are disfavored reactions, which does not happen in standard organic reaction, which that you can force by catalytic antibody, you can force the reaction to follow a disfavored pathway. How? So, let us take this example. This is a phenyl with some substituent, then CH2 and an epoxide, and then the epoxide is connected ultimately to a hydroxy group. Okay. Now, here if you add some acid, so this will be protonated, the epoxide, and as soon as it is protonated, because epoxides are strained molecule, they will open. Okay. How they can open? Their opening is assisted by this adjacent nucleophile, which is OH. Okay. So, basically I can write it here. So, now you have two modes of ring opening. One is this can attack here, that opens up, or the other is this can attack at this carbon and this opens up. Okay. So, these are the two possibilities. Now, one is called 5-exotate. 5-exotate. What is tate? Tate means you are attacking at a tetrahedral carbon, because these are tetrahedral carbon, sp3 hybridized. So, tate. Why it is called 5-exo? Which is 5-exo? That means when it is attacking that one, what you are making is a 5-membered ring. So, that is 5, and your whole epoxide is outside the ring that is being formed. So, that is why that is called exo. So, this is 5-exotate. The other possibility where the lone pair attacks this carbon, now this bond containing the epoxide becomes a part of the ring. So, this will be called endo, but again you are attacking to a tetrahedral carbon, but this is called, as you are making a 6-membered ring, this is called 6-endotate. Okay. So, this is 6-endotate product, this is 5-exotate product. Now, according to Sir Jack Baldwin, there are some rules called Baldwin's rules, and if you do this reaction, you will see that this is the product which is formed because Baldwin's rule says that 5-exotate is favored over the 5-exotate is favored and 6-endotate is disfavored. Okay. So, in the test tube, if you do this reaction with the chemical reagent, it is acid, say like some Lewis acid, you will get this product as the measure. But if you want this as, if this your target is to make this compound, then the question is how to make a disfavored reaction favored. Okay. So, what was done is that when this compound, when this is attacking in the disfavored reaction, which is the endo, when it is attacking, so part of it is now broken, part of this CO bond. So, this will be delta minus and this oxygen will be delta plus. So, the intermediate or the transition state, maybe I can draw the transition state. So, that is delta minus, and then what you have is oxygen. These are half bonds. Okay. These are not formed fully, and this is the aromatic ring, and that will be delta plus. So, that will be the, if you can mimic this with a molecule, then that will be your hapten, and with that at hapten, if you can generate a catalytic antibody and give it to this system, then that will do this reaction. Then that will do the 5-6-endotate. Okay. So, what actually resembles this compound, because this is not stable. So, finally, they come out with a molecule which is an N-oxide molecule. Now, look at this molecule and try to check here. See, what you need is a carbon here, which is it may not be carbon, you can have different atoms, but it should be attached to a kind of a negatively charged oxygen. So, that what you have, instead of the carbon you have a nitrogen here and you have a O minus here. Okay. But what you needed is a delta plus center at the next carbon, that means here you want a delta plus here and you want a minus here. Now, because this nitrogen is positively charged N oxide, so via inductive effect this will generate a delta plus at this position. So, now if you look at this one and that one, they resemble each other by, say, this is tetrahedral, that is tetrahedral, this has got a negative charge, this is the negative charge, and this is the positive charge, and this positive charge is generated by inductive effect, and this R is utilized to attach the protein. Okay. So, now if you can generate a catalytic antibody against this, and that will act as a catalyst to do a disfavored reaction, and ultimately you get this product. In fact, this was done, and this was a very classic example, and it showed the power of catalytic antibody to do unfavored reactions. There are many more examples of unfavored reactions by using catalytic antibody approach. We do not know any other approach by which you can get a disfavored reaction into a favored process. Okay. So, I think that ends this this aspect of synthetic biology, that is the enzyme chemistry or the catalytic antibody chemistry. Okay. Thank you.